Staphylococci. Staphylococci are gram-positive cocci. Gram-positive pathogens can be divided into two categories: gram-positive cocci and gram-positive bacilli. Gram-positive cocci include staphylococci, streptococci, enterococci. Gram-positive bacilli include Bacillus corynebacteria, Clostridia, Mycobacteria, Actinomyces, etc. Cell division can occur in two different manners. Either it can occur in one plane or in three dimensions. If the cell division takes place in a single plane and the daughter cells remain together, it will give rise to a chain of cocci, which is known as streptococci. And when cell division takes place in three dimensions and the daughter cells remain together, it may give rise to a cluster formation, which is known as a grape-like cluster. And staphylococci occur in grape-like clusters. Staphylococci, the word is derived from the Greek word staphyle, which means bunch of grapes. They are gram-positive cocci arranged in clusters. They are hardy organisms which can survive in adverse conditions as well. Major human pathogens Staphylococcus aureus and several other skin commensals are included in this group. The family Micrococcaceae has genus Micrococcus and Staphylococcus, which have more than 20 species, including Staphylococcus aureus, Staphylococcus saprophyticus, Staphylococcus epidermidis, etc. They are gram-positive cocci, non-motile, facultative anaerobes. Cells occur in grape-like clusters because cell division taking place in three dimensions and daughter cells remaining together. Staphylococci are salt-tolerant. They can tolerate higher concentrations of salt. They are even tolerant to desiccation, that drying of environment. They can survive on fomites because of this. These are cocci in clusters, gram-positive cocci in clusters as seen in the microscope. Many species are medically important in Staphylococci, out of which three common species include Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Staph aureus is the most virulent of all of them. It causes bacterial infections, it causes food poisoning and toxic shock syndrome. Staph epidermidis is an important cause of prosthetic implant infections and endocarditis. Staph saprophyticus causes urinary tract infections. It causes cystitis, especially in women. This is a slide which shows a sample of a patient stained by gram stain, which shows pus cells. There are plenty of pus cells and there are gram-positive cocci in clusters. Patient has a staphylococcal infection. We can divide staphylococci into two groups: coagulase positive staphylococci and coagulase negative. Coagulase positive includes Staph aureus and coagulase negative include all other staph, including Staph epidermidis and saprophyticus. Staph aureus is a major human pathogen habitat. It is part of normal flora on the human body, in the nose, in the perineum. Source of organism can be a human carrier, it can be a patient, it can be environment, it can be fomites. Staph aureus is a facultative anaerobe, grows at 37 degrees Celsius, pH 7.4. Media useful for Staphylococcus include nutrient agar, blood agar, McConkey agar, and several special medias. On nutrient agar, Staph aureus shows golden yellow pigment, which is non-diffusible in the medium. Staph epidermidis and saprophyticus either do not show pigment or they may show white colored pigment. On blood agar, Staph aureus shows beta hemolysis, while epidermidis and saprophyticus do not show hemolysis. Special media like mannitol salt agar and Ludlam's medium can be used for Staphylococcus aureus as selective medias. The figure shows colonies on blood agar which are beta hemolytic, and another figure shows mannitol salt agar, Staphylococcus aureus grown on mannitol salt agar. In biochemical reactions, Staph aureus is catalase positive, and all the Staphylococci are catalase positive, and all the Streptococci are catalase negative. So this property, catalase test, is used to differentiate between Staph and Strepto. Staph aureus is also mannitol fermenter. It is phosphatase positive. In the catalase test, hydrogen peroxide is used, and prompt effervescence indicates catalase positive. No effervescence indicates catalase negative. You can also perform the catalase test on a petri dish. Here you can see catalase positive test in the petri dish and another one catalase negative. Coagulase test में fibrinogen is converted to fibrin and the fibrin leads to clotting of plasma. Staph aureus has coagulase enzyme which leads to coagulase test positive. The clotted plasma will not move in case of coagulase positive, and in case of coagulase negative, the plasma will freely remain as a fluid. There are several signature features of Staph aureus which include coagulase positive, mannitol fermentation, phosphatase production, reducing telluride, thermostable nucleus production, beta hemolysis, golden yellow pigment formation, and gelatin liquefaction. Staph aureus is a skin commensal found in various human population, in carriers, in the nasopharynx, in the perineum, and people can shed Staph aureus from their secretions, from the nasal secretions, and it can be acquired by direct contact or through fomites, by dust or by airborne droplets. Staph aureus is a common cause of post-operative wound infections and other hospital cross infections, and various multi-drug resistant strains of Staph aureus are prevalent in the hospital setup, which gives rise to multi-drug resistant staphylococcal infections which are difficult to treat. Staph infections result when staph they breach the body's physical barriers, and even a few hundred of cocci can enter the body and give rise to disease. Staph produces several enzymes and toxins which act as virulence factors for it, and it has various mechanisms by which it can evade phagocytosis. Structural defenses of Staph aureus against phagocytosis include protein A, which coats the cell surface and it inhibits complement cascade and interferes with humoral response. The clumping factor or bound coagulase, which gives rise to blood clots, and these fibrin clots they hide the bacteria from the phagocytic cells. It synthesizes loosely organized polysaccharide slime layers known as capsules, which inhibit chemotaxis and phagocytosis. Staph aureus produces various enzymes like coagulase, hyaluronidase, staphylokinase, and lipase and beta-lactamases, which are various virulence factors of this organism. It produces bound coagulase and free coagulase, two kinds of enzymes. The bound coagulase enzyme is responsible for slide coagulase test, and the free coagulase enzyme is responsible for the tube coagulase test. In the tube coagulase test, the free coagulase does clotting of plasma with the help of the factor known as coagulase reacting factor. Various toxins produced by Staphylococcus aureus include cytolytic toxins, exfoliative toxins. The exfoliative toxin cause the patient's skin cells to separate from each other and slough off the body. Cytolytic toxins, exfoliative toxins, toxic shock syndrome toxin, and enterotoxins. The enterotoxin will be responsible for food poisoning. The toxic shock syndrome toxin will be responsible for toxic shock syndrome. Various virulence factors of Staphylococcus aureus can be depicted in a pictorial manner like this. Staph aureus being a commensal in the human body, skin, in the perineum, nasopharynx, etc. And it can lead to abscess formation in different sites. It can lead to various diseases due to toxin production. The two kinds of diseases are produced by Staph aureus, those due to direct effect of the organism and those because of the toxin. Those which due to direct effect include abscess formation, local lesions of the skin, and systemic infections. And those which include because of the toxin include food poisoning, toxic shock syndrome, and scalded skin syndrome. Staphylococcal disease can be divided into three categories: the non-invasive diseases like food poisoning, cutaneous diseases like impetigo, skin syndrome, folliculitis, etc., and systemic illnesses like bacteremia, endocarditis, pneumonia, osteomyelitis, and toxic shock syndrome. Various diseases caused by Staph aureus can be depicted like this in a pictorial manner in one photograph. Factors predisposing to Staph aureus infections include host factors and pathogen factors both. Various factors on the side of the host can also facilitate staphylococcal infections like breach in the skin, chemotaxis, opsonization, neutrophil functional defects. diabetes mellitus, defects of opsonization and chemotaxis, presence of foreign bodies that can help or facilitate causing staphylococcal infections, and several factors on the side of the pathogen like catalase, coagulase, hyaluronidase, lipase, etc., beta-lactamase, they will also contribute to causing infection. Skin and soft tissue infections caused by Staph aureus include folliculitis, furuncles, carbuncles, abscess, impetigo, wound infections, etc. Deep localized infections like osteomyelitis. Staph aureus is the most common cause of osteomyelitis. It can also lead to arthritis. Respiratory tract infections include bronchopneumonia, tonsillitis, pharyngitis, sinusitis, etc. These are various photographs of staphylococcal lesions. CNS infections like abscess, meningitis, intracranial thrombophlebitis, endovascular infections like bacteremia, septicemia, pyemia, endocarditis, urinary tract infection, nosocomial infections can be caused by Staph aureus, staphylococcal food poisoning because of the enterotoxin produced by Staph aureus. It occurs about 1 to 6 hours after consumption of the food. So this incubation period of 1 to 6 hours is important to be noted, because for different food poisoning causing organisms, the incubation periods are different, and the incubation period can give a hint towards the causative organism. So remember that staphylococcal food poisoning incubation period is about 1 to 6 hours, and type A enterotoxin is most responsible for the cases. Then toxic shock syndrome caused by toxic shock syndrome toxin. It was discovered in tampon related cases, and it is a fatal multisystem disease. It has been mostly in females, but less commonly even in the males it has been observed. The toxic shock syndrome toxin causes this toxic shock syndrome. It was discovered in cases of tampon users, तो उसमें से उसका identification हुआ. Various signs and symptoms of toxic shock syndrome include nausea, vomiting, watery diarrhea, high-grade fever, prodromal period, mucosal hyperemia, headache, myalgia, confusion. Skin rashes starting from the trunk and then spreading to the periphery. Staphylococcal scalded skin syndrome, which is caused by exfoliative toxin. The severe form of staphylococcal scalded skin syndrome is Rita's disease in newborns and toxic epidermal necrolysis in the older patients, and the milder forms are pemphigus neonatorum and bullous impetigo. Staphylococcal scalded skin syndrome in small children, it can lead to this kind of a picture where there is generalized erythema, fever, and skin slipping off with gentle pressure, that is Nikolsky sign, exfoliation or desquamation of skin, and it's a painful condition. Treatment is fluid rehydration and topical wound care, and it is a self-limiting condition in which a full recovery is expected. And possible complications can be septicemia, cellulitis. Lab diagnosis of staphylococcal infections includes specimen collections, specimen transport, and then processing. Collection of specimen depends on the type of lesion: pus in case of suppurative lesions, sputum in respiratory tract infections, feces and remains of food in case of food poisoning, blood in case of septicemia, nasal swab for detection of carriers. And after collection of specimen, it is transferred to the laboratory as fast as possible. And in the laboratory, microscopy can be done initially, which shows gram-positive cocci in clusters in the gram stain, growing on nutrient agar, blood agar, McConkey agar. Selective media like Rodham's medium mental salt agar can be done. Biochemical tests like catalase test, coagulase test can be performed, and various battery of tests or signature features of Staph aureus that were mentioned earlier, they can be used as test for identification of Staph aureus. Various coagulase negative staphylococci are there, like Staph saprophyticus, epidermidis, etc. General features of coagulase negative staph include that they are important agents of nosocomial infections, and they have been associated with infection of implants such as CSF shunts, IV lines, cardiac valves, urinary catheters, artificial joints, etc. The morphology is similar to Staph aureus, however, they form white colonies or are not forming any colored colonies, and they are coagulase negative. Staph epidermidis colonies white in color. It is also gram-positive cocci in cluster. Diseases caused by coagulase negative staphylococci include prosthetic valve endocarditis, meningitis, peritonitis, UTI in pregnant women caused by Staph saprophyticus. It can be treated with vancomycin if not resistant. Staph saprophyticus responds to trimethoprim or to quinolones. You can compare the various features of different species by forming tables, and in that you can enter the important characteristic features of the organisms like coagulase test and mantle fermentation. In case of Staph aureus, they are positive. Then to differentiate epidermidis from saprophyticus, novobiocin test is used. Epidermidis is novobiocin sensitive, while saprophyticus is novobiocin resistant. Antibiotic sensitivity pattern is quite variable. Various enzymes or methods of resistance to drugs have been developed by Staphylococcus. Beta lactamase production, which is plasmid mediated, and alteration of penicillin binding proteins, which is chromosomal mediated. These are mechanisms for drug resistance found in Staphylococcus aureus. Testing is done by using methicillin in the laboratory, methicillin resistant Staphylococcus aureus MRSA, which is an emerging problem in the world over. Another term used here is VIRSA, now the vancomycin intermediate resistant Staph aureus, or even VRSA, vancomycin resistant Staph aureus. Treatment of staphylococcal infections can be based on the type of infection and drug sensitivity report. Vancomycin and other drugs can be used. Drug resistance in Staphylococcus aureus is an important problem in nowadays because drug resistant strains have increased, and in the hospital setup many of the strains of Staphylococci are multi-drug resistant, leading to very serious infections in the patients. And there are various methods by which the organism can develop resistance to penicillin: mutation in the chromosomal genes, acquisition of resistant genes as extra chromosomal plasmids, production of penicillinase, development of tolerance, and changes in the bacterial surface receptors. So these are different methods by which the organism can develop resistance to drugs. MRSA is methicillin resistant Staphylococcus aureus. There are two types of MRSA: community acquired MRSA and hospital acquired MRSA, CA-MRSA and HA-MRSA. The hospital acquired MRSA is much more serious and it is resistant to many other drugs as well. And hospital personnel, they can also act as reservoirs of MRSA because some people, they harbor MRSA in their nose as commensals. MRSA can be transmitted through hands, especially of healthcare workers. Infected patients or carriers can harbor these organisms and transmit to other people. It can be found on devices and various items or environmental surfaces also. Treatment of MRSA, vancomycin teicoplanin can be used. Another term, that is VRSA, vancomycin resistant Staphylococcus aureus. VISA, VRSA, MRSA, these are various terms used for drug resistant Staphylococcus aureus. For prevention, various measures can be done like control of nosocomial infections, aseptic techniques, etc. This is a case study in which a 30-year-old female was brought to the hospital. She had fever, erythematous maculopapular rashes over her body, and she was confused. She had a history of high-grade fever, nausea, vomiting, and deterioration in her condition over the last 24 hours. The menstrual history, she is on day 4 of a cycle. The parameters have been shown. The buccal mucosa and tongue are congested. Tampon in place when removed reveals a red and inflamed vaginal mucosa. The blood picture shows leukocytosis, thrombocytopenia, abnormal liver function test, and elevated urea and creatinine. Specimen vaginal swab, tampon and blood, microscopy में gram-positive cocci, culture में beta hemolytic colonies of Staph aureus on blood agar. Biochemical test में catalase and coagulase positive and blood culture positive. The causative agent was Staphylococcus aureus. The diagnosis was toxic shock syndrome. And diagnostic features include high fever, skin rashes, multi-system involvement, inflamed vaginal mucosa with tampon, and microbiological findings. With this, we end this session. Thank you.